Good morning, friends. This is Vinoy Krishna, and we are going to join together to discuss the Nobel Prize Chemistry for 2024 this year. Just as we had the discussion on the Nobel Prize in Physiology or Medicine the last day. Interestingly, quite similar to the Nobel Prize in Physiology, this year's Chemistry's Nobel Prize also has a very close link and a very overlapping applications with one of the already awarded Nobel Prize in 2018. So let us begin with this very question that at the end of the discussion we would try to address and see how the sciences from different directions, they all may have the merger at one common goal. Right. So what is common between the two Nobel Prizes? The one that has been awarded in 2024, chemistry, to three people, one half going to the American David Baker, University of Washington, for his categorical work, that is computational protein design. Based on the computational abilities of the software programs, you will be able to engineer the novel proteins for multiple applications we'll try to focus on. And the other half goes to a couple of corporates from United Kingdom, Demis Hassabis and John M. Jumper. They have been awarded the Nobel for protein structure prediction. We will get to see how this is one of the biggest challenge since the biology started getting molecular at its level. So in a way, this has been a huge achievement or a watershed moment, you could say, in molecular biology in general and proteomics in particular. So let us see what have these people done, and we will later see how their work is quite comparable, quite overlapping with the work of Frances H. Arnold, who was awarded the Nobel Prize in 2018 for her directed evolution of enzymes, and how both of them overlap functionally. Just few background fundamentals to begin with. Kind of a cliched thing now, all of you, I hope, already know that the entire genetic material having the information within the cell present as the DNA. The genes on the DNA get decoded in their information through the first copy called the RNA through transcription, and this RNA ultimately, based on what sequence of bases it has, gets to decide the protein construction. People, the protein is nothing but a polymer of amino acids. Amino acid is one of the common chemical molecule present in the biological systems. Now, based on this RNA, you create multiple amino acids to be tailored or stitched together into a chain of amino acid, and that chain of amino acid is what you call as polypeptide or protein. Long story short, remember that proteins are the most versatile of all the molecules that we have in the biological kingdom, and it is because of these specific proteins that we have in our cells and all around our body, we are able to function the way we do. You are able to hear me out, you are able to see the screen, all because of the dedicated proteins which are in a team involved in giving you those physiological abilities. So let us come back to the protein. Just a quick glance at how at the different levels the proteins perform different functions for you. So at a macro level, you can understand your muscles are primarily the proteins which give the muscles the ability to contract and relax, the contractility as you call it. Further, there are many hormones, not all of them. There are many hormones like insulin, the growth hormone, which are also polypeptide or proteinaceous in nature. Antibodies, the immunoglobulins we so badly depend upon whenever our body is attempt invaded by the pathogens, right? Hemoglobin, such an important protein that all of you know about, the protein which transports oxygen all across your body through your blood. And then there are many. The cells have on their surface the receptor, the major group of proteins called enzymes, people. Enzymes, I hope you know, are the biocatalysts, what we call as life, or that giant embodiment that we represent as a person. We are a huge laboratory, and within this laboratory, at any point in time, whether you are active or resting, millions of or lakhs of reactions which are happening, and all these chemical reactions are catalyzed by the dedicated proteins called enzymes. So what we now know, that almost every function of the body is dependent upon these important molecules called the enzymes, the proteins, and the proteins exist in the different formats. Now, because they are so important, the immediate curiosity is what makes these protein be so versatile to give you all those functions. People, in the natural systems, there are 20 different standard amino acids. So all the proteins are made up of the 20 different amino acids to be used in all possible sequences that you can think of using those amino acids. Just as using 26 alphabets in the language, you can create infinite number of texts and the messages. Fine. So what we now have is the amino acids form the chain. Ultimately, what are they? Chemical molecules. Every chemical molecule is characterized by the individual charges. Think of your school days when you learnt about the different forms of chemical bonding, wherein covalent bond and non-covalent bond. So what we understand, based on the sequence of amino acids that are there in the protein, the chemical interactions will happen, and based on chemical interactions, these amino acid chain might acquire a certain shape. Can you see? This is an image which is trying to simplify that for you. It is each of the circle is an individual amino acid out of the 20. So all random sequences that are possible will, based on the sequence of amino acid, create a particular kind of looping. What you're getting to see in this image is popularly called as alpha helix, or here also like a plain ribbon kind of a structure. So beta pleated sheet it is called. So what we now know, that based on the sequence of amino acid, the chain may fold itself into a structure like helical or plate-like, and that structure is what we call as the secondary structure of the protein, which means what is the primary structure? The primary structure refers to the sequence itself. Could you visualize the old days telephonic cables, the PNT phone? I mean, those were the days when the landline connections, the phone used to have the connection of the wire that had the capability of looping in a spiral manner, and then that helical thing also further multiple times twisted in itself. Can you see this structure? So what we now have to understand, people, that this secondary structure that has been created will multiple times fold on itself as well to create very complex three-dimensional structures of protein. These three-dimensional structures are called tertiary structure. And take it as a simple philosophy, not necessarily the abstruse science here. Everything in the world which is physical, has got its function directly guided by the structure it has. The chair that you might be sitting on right now, or the pen that you might be holding in the hand, or anything that you are using has got a function which is directly derived from the structure it holds. Exactly like that, the functions of the protein is derived from the structure the protein has. Having understood that the structure of the protein primarily at three levels: primary structure, which refers to the sequence; secondary structure, based on the sequence it takes into a specific looping pattern; and then based on the sequence again, further multiple foldings into the tertiary structure. Can you get it? Something very much similar to how you make your sweater starting from the wool or the cotton. So based on the wool that you have taken from the sheep, you may turn it into the yarn, and these yarn are further folded back into a particular pattern to create the functional structure. This, people, in that analogy would refer to the tertiary structure of the protein. Now listen carefully. Given the importance the structure of the protein has for its function, you see, in 1972, there was this fellow called Christian and Vincent. He made this first observation and conclusion that the structure, 3D structures of the protein and hence their function, is directly decided by, encoded by the sequence of amino acids. So he made that connection. And how did he do that? Because every time he was trying to, stretch and open up, let us say when you heat it, or you could visualize you're physically stretching, the protein has the tendency to get back its original shape, which means it is in the sequence to get back to that structure. Now, the moment he brought this out, he had made a huge revelation, and that gave him the Nobel Prize for 1972. But then he had just initiated a line of thought. Can you, continuing with the same understanding, visualize these toys of the kids, wherein you have those blocks interconnected which you can fold into different shapes. What you have to realize, of all the possible shapes you could fold them into, imagine for any protein, any one particular shape is what is the functional shape, and all others which in different conditions could be created will be called as non-functional shapes. And in those shapes, the very same protein that were giving you certain functions will now start spoiling it, and hence become the reason for many diseases. That is called as misfolding of proteins. And you would see that there are many diseases that you know of are possible because of the protein misfolding. So what is it? Like Alzheimer's, like Parkinson's, like Huntington's, like Creutzfeldt-Jakob disease, like cystic fibrosis, ALS, many others. So I hope you are now getting into the line that understanding the protein would also help us understanding the various anomalies, and maybe find out the ability to diagnose it as well as to cure it. So that is the prime basis. But the question is, how could we study the structure of the protein? Because it is extremely difficult. These are micromolecules. I mean, they are very small in size, and they are very delicately balanced in that structure. And that is why people, over the decades, there have been many technologies that have come up, all trying to study the different proteins of our body. There are thousands and thousands of different proteins that a body has. Like, electron microscopy based, like X-ray crystal, just, the name familiarity is enough for your requirement. So electron microscopy based, so you observe the, under the microscope. X-ray crystallography, you fire X-ray on the protein, and based on how the X-ray gets scattered, you will find out through reverse engineering the structure of the protein. Or people, a very popular method called nuclear magnetic resonance spectroscopy. You need not get into the details of any, but just to tell you, all these conventional methods of protein structure, given the importance of the topic itself, went on to win the Nobel Prizes in the respective year. 2017, 1964, 1952 for the microscope, crystallography, NMR. But then there is a problem. All these methods, they take a huge lot of time. Over a decade maybe, to study a single protein. And now come to think of this. Today, we have a sequences of the amino acids of around 200 million different proteins. But out of those 200 million different proteins, given that these conventional methods, they are so slow, they are so taxing, they are so costly, that we have the protein structure of just around 2 lakh proteins. Way off the mark, the demand supply. And that, people, was called as the 50-year-old grand challenge in biology by our Indian origin scientist, another Nobel laureate, Venki Ramakrishnan, who won the Nobel Prize for his diligent work on decoding the structure of ribosomes. Now, when we look at this present year's Nobel Prize, the work of Venki Ramakrishnan would now start appearing to be much more hard work than what today it would take. But yes, he deserved it, and he called this problem as the 50-year-old grand challenge in biology. And that is why the Nobel Prize of this year gets justified. So let us come back to the heroes of this year. People, one half of the award goes to this fellow of University of Washington, David Baker. Now what he did, people, there is this, biannual competition on a global basis that happens called Critical Assessment of Structure Prediction, wherein the scientists from across the world sit for that competition, use all the, multiple computers, and through their software try to decode the protein structure. So Mr. Baker was a regular participant in that CASP event, and he ended up developing a program called Rosetta. This program name could be important for the exam, please remember. The Rosetta computer program. This program allowed Dr. Baker to study the already known structures, and based on that combine the individual structures of different proteins and then make up a novel protein altogether. A protein which is naturally not seen, with a specific structure. And if the structure has function, obviously with a new function as well. In doing that, Dr. Baker initially first created a protein which the structure that Rosetta program said was confirmed by the conventional method, thereby spelling the success of the program. And later he also did prove that through this method they could create new enzymes, new structures could be generated by following the already available ones, and they could generate new enzymes, and these enzymes could have novel functions. I'll take you through all these possible functions. Expect straightforward question on the functions or applications of protein engineering or synthetic biology. So they suggested that okay, it is possible to create multiple enzymes having the different functions. So let us now concentrate on, based on Baker's discovery that we can create new proteins by following the examples from the already available one through the capable software program, the Rosetta program as we called it. So he or his success, you can see today, has got multiple applications. For example, you see industrial applications. As we go on to produce new enzymes or new proteins with new functions, they can give you functions in many different areas. For example, the biofuel production. You can connect it with the Bio E3 policy of the government, wherein we want to become the global biomanufacturing hub of the world. So biofuel production, particularly to make our energy consumption more sustainable and more environment friendly. Yeah, food processing. You'll be able to use these enzymes for generating better food products, better processing, lower wastage, better flavor, and multiple other biological products that I'll also give you a brief hint of. Yeah, it could also be used to create new proteins for researchers to understand the proteins in a little better manner, which may in future may have furthermore applications. People, we can use the protein designed in the lab or developed in the lab also to study the various diseases, and then use those proteins that we have produced in the lab to detect the diseases also. So those proteins could be used for detecting the diseased status within the particular cells. People, the market of production of biopharmaceuticals. You might have heard about monoclonal antibodies, the different hormones like insulin, growth hormones, for example, the clotting factors, and many other enzymes which are important for therapeutic application. All of these are called biologics, and these biologics are today ruling. You can understand the protein-based drugs market is today valued at nearly $400 billion. And these proteins could be created through the help of very quick development of the structure on the program without having to go through the rigors of the lab in the conventional route. Fine. And then something that you can immediately connect with. I believe, given the current importance, all of you have studied about the CAR T cell therapy. The T cell, which is one of the potent immune cell of our body, is attached with the specific receptor protein. That receptor protein has also been lab engineered, people. So that lab engineered protein on the T cell helps the T cell to bind and particularly kill the cancer cell. So you can understand it opens up many new vistas of applications, and that is where the importance of Baker's work and why he has been awarded with the Nobel Prize. Followed by the other half of the prize that goes to two of the fellow mates, Demis Hassabis and John M. Jumper, where these people in 2010 they created the company called DeepMind, and this company uses the computer-based programming to convert the sequence of amino acid into the most possible natural 3D structure it would take up at the, using the deep learning or neural network, which is directly, following the neuronal connections that we have in our brain also. So these people developed the program called AlphaFold. And this tool, AlphaFold, when given a particular amino acid sequence, immediately converts it into the most possible natural 3D structure it would take up. AlphaFold initially could not, achieve success beyond 60%, but in the course of time, they have further through the, machine learning, they have improved upon the AlphaFold, and the new version of AlphaFold, AlphaFold 2, is capable of, given any amino acid sequence, converting it into the structure in the simulation, which is 90% plus similar to what we achieve through the conventional method, crystallography, NMR, or electron microscopy. And this, people, is a huge, huge success. Mind it, you remember 200 million sequences available with only 2 lakh the proteins that we have been able to structure through the conventional method? Look here, the day AlphaFold gets into action, it has, in a matter of few hours, converted all these 200 million, sequences that we have into the most possible structures of the protein. So that 50-year-old grand challenge has kind of been surmounted, or you have won over it. And that is going to open up the world of possibilities of applications. You see, this AlphaFold 2 is capable of, so for all these screens that I'm showing, in order to take the notes down, you can just get a pause and take it down, as these could be important. So you see, the prediction of the possible structure could then help you in multiple different ways, which is exactly overlapping with what you have already studied in the synthetic biology, with Baker's case. So with more knowledge of the proteins, you'll be able to do structural biology better. You'll be able to understand how the different proteins in the cells interact with each other. The mechanism of action, how the protein acts, because every protein binds to something in exactly the same manner as the lock binds to the key. So knowing the structure of the key and the lock would be helpful in decoding the minor aspects of the cell function, and hence use it to our advantage, right? You can also study the protein evolution by comparing the different proteins of the different organism. You'll be able to understand how the proteins in humans could be better, or sorry for the wrong term here, could be different than the protein of any other organism that you're comparing yourself with. So these structural comparison and how we are evolving could also be understood. And most importantly, people, we'll be able to work out the proteins which could be again used as the drugs for the treatment of the rare diseases, the development of protein-based vaccines. So all of these possibilities suddenly get created. And then there are other subsidiary advantages also that you can mention in the exam, like the availability of these tools will certainly enhance the biology and bioinformatics curriculum. Our students would be able to learn the better realities of it, something in the classroom itself, and that will further help in the future technologies that will be developing. I have already talked about the different diseases treatment and the food processing, the textile industry, the paper industry, everywhere where you require the enzymes, people. So what we now understand that both these works are kind of interconnected. One is predicting the possible structure, another is talking about creating a new design. But then both will help each other in a complementary manner. But then, just as any technology, this as well would be value neutral, and hence would always leave the scope for some of the risks or the hazards based on the intention of the person behind it. So what are the challenges associated? Could you see that in some of the cases, you could use this to generate the new versions of the pathogens with the proteins in them much more, capable for increasing their pathogenicity or virulence, and could be used to augment the particular agents or entities' ability to launch the bio-warfare. And hence that could be one of the major lacuna that could be there. Further, you can think of creating new toxins, and those toxins could be again used for, proteins could be toxins, could be used for multiple malefied intentions as well. So yes, while the technology evolves to give you superpowers, at the same time the evolution of the governance or the regulation has to keep pace, so that we utilize the positives of it without falling prey to the negatives. Right. Now, before we end the talk, just to leave you with what was the Nobel in 2018 given for and how they are interconnected. Bachcho, the 2018 Nobel Prize was given for directed evolution of enzyme, which is also largely about synthesizing new enzymes only, but that depends upon a different route of it. What the lady did, took the gene coding for a specific enzyme and conducted random mutations in it, random changes in it, and each of the mutated gene is introduced into the different bacteria, and these bacteria, as they express the gene and produce the different versions of the enzymes, they check whether any better enzyme has been created than the natural version or not. So yet again, this also, through this route, was about synthesizing new proteins better than the earlier ones, serving the similar purpose. Just the route was through random hit and trial, and what we now have awarded was for computer-based simulation, which is being utilized for that. So yes, the different Nobel Prizes serving the same purpose, and that makes, interestingly, both these Nobel Prize winners quite connected in the ultimate purpose they serve. I hope you are able to understand it. The topic would remain important for both prelims as well as the mains, so you can take down the notes from this. Hope to see you soon. Bye-bye.